skeletal muscle cells cannot stimulate themselves. To be stimulated or excited, they must be signaled by neurons, specifically motor neurons, which are neurons or nerve cells that stimulate muscle cells or fibers. This occurs at an area called the neuromuscular junction. This is an example of a synapse, which is a broader term meaning the junction or meeting area between a neuron and some other cell. A synapse could be between a neuron and another neuron, a neuron and a gland that secretes things like hormones, or a neuron and a muscle cell. And it's these last synapses that are termed neuromuscular junctions. So neuro for neuron, muscular for muscle, junction just means a meeting area. A motor unit refers to one motor neuron plus all the muscle fibers it controls. So it's kind of like when you have a sled that's drawn by huskies, sled dogs. You have one sled with a person on it that's controlling the dogs, and there's multiple, many, 10, 8 sled dogs that are pulling the sled. They're moving, but they're all responding to the commands of the one person that's driving the sled. So the neuron, the motor neuron, is the person, and the muscle fibers are the dogs. In this example, you see 3 neurons. They're color-coded, and each neuron is controlling a set of these muscle cells. As you can see, the muscle fibers are interspersed within the larger muscle organ, and this is how our muscles are really organized. Motor units vary quite a bit in size, with our smaller muscles that are capable of finer movements, like the muscles of the eye, having only a few muscle fibers per neuron, 2 or 3. Our larger muscles that are capable of powerful movements but are not under such fine control can have thousands of motor fibers per motor neuron. The muscles of our quads, like the rectus femoris, the thigh, has thousands, 4, 5, 6,000 muscle fibers per neuron. So varies tremendously depending on the function of the muscle and the size of the muscle. When we zoom in on that neuromuscular junction, we're going to see specific areas. And these areas are important to understand how an action potential travels from a neuron to a muscle cell, how our nervous system signals our skeletal muscles to contract. Zooming in, we have both a presynaptic terminal and a postsynaptic membrane. The presynaptic terminal is the neuron side of things. Pre for before. This is before the synapse or meeting. The very end of the neuron, called the axon, that travels away from the body of the neuron towards the muscle cell. Quick recap from chapter 4. Recall that neurons have this really characteristic structure, which we will talk about more in chapter 8, but we talked about briefly when we explored tissues, where there's multiple short tapered dendrites, and then our cell body is the biggest part that contains a lot of organelles like the nucleus. And then extending away from the cell body, we have this axon. A long process, usually singular. Action potentials or signals travel in just one direction from dendrite to cell body to axon, and then there's some other cell here, be it another neuron, a glandular cell, or a muscle cell that the neuron is talking to. So here we're just right here at the presynaptic terminal. The postsynaptic membrane is the muscle side of things. So it's that specific portion of the sarcolemma or cell membrane that interacts with the neuron. It looks different from the rest of the sarcolemma. It has a creased or folded cell membrane that increases the surface area, making more room for receptors that can receive a signal from the neuron. So it kind of looks like this. Just a lot of creases or folds to increase surface area. You'll see it referred to as the motor end plate. Motor end plate. These two cell membranes, so axon cell membrane of the presynaptic terminal, the sarcolemma cell membrane of the postsynaptic membrane, they're separated by a very narrow space called the synaptic cleft. This means that the axon and the muscle cell don't actually physically touch, and this maintains a level of control to the nervous system. Well, it poses a problem though, right? Because if the cells don't touch, how does that electric action potential, that depolarization, travel from cell to cell? Kind of a problem. Well, it means that another type of signal is necessary here. For a non-biological example, think about when we travel around the city. If you are like me, I used to teach on the West Bank, but I live over on the East Side. Not ideal. So I'd have to drive to the Crescent City Connection and cross that, and then go on General Meyer and get to the West Bank campus. One time, it was a Saturday, so I left late because traffic is usually really minimal. The whole bridge was shut down. Now, had I left earlier, had I been prepared and checked the traffic, what could I have done? I could have taken the ferry, right? Well, probably not because everyone else was trying to take the ferry, but in a perfect world, I could have taken the ferry. So, when you take the ferry, you drive to the ferry terminal. Then you have to change your mode of transport and get on a boat, which carries you across the river. Then you can get back in your car and drive to wherever you needed to go. This is like what happens here at a synapse, like the neuromuscular junction. We have an electric action potential traveling down, down, down the axon till it can't go anymore. It's reached that presynaptic terminal for end, so very end of the axon. Can't go anywhere. That is going to stimulate another type of signal, a chemical signal, to be released via exocytosis. Remember, exocytosis is when a cell releases a large amount of a substance through vesicles. Vesicles that contain something fuse with the cell membrane and dump their contents out of the cell. In that presynaptic terminal, we'll see special vesicles called synaptic vesicles. Remember, a vesicle just means a small membrane-bound sac that has something in it. It's just called a synaptic vesicle because that's its location. It's found here at the synapse. It's the only place it's found. The purpose of these vesicles is to store chemicals called neurotransmitters. These are specific chemicals that the neuron produces that can either stimulate or inhibit the postsynaptic cells it's signaling. Neurons typically have a couple of these that they use. Any given neuron will have 2 or 3 neurotransmitters it's using to signal its cells. These can either increase the potential of an action potential occurring on the postsynaptic cell or take it further away, inhibit signaling of the postsynaptic cell. There's a lot of them, but there's only one used here at the neuromuscular junction of skeletal muscles. That neurotransmitter is called acetylcholine. Shorter abbreviation is ACH, because acetylcholine is a little cumbersome to write and to spell. It comes from the acetic acid, choline for the choline group that come together to form it. So, acetylcholine is the neurotransmitter that's stored within the synaptic vesicles of the neuromuscular junction, within the presynaptic terminal of the axon. When an action potential travels down the axon and hits the presynaptic terminal, the synaptic vesicles will fuse to the cell membrane, dump out that acetylcholine. And acetylcholine then, like the ferry, is going to drift across the cleft and signal the postsynaptic cell. Signal our skeletal muscle cell to contract. So this is our muscle fiber. This is the axon of our neuron. So this would be our neuromuscular junction. We're zooming in on it here, looking at just one presynaptic terminal. And we can see action potential would go down, 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 down here, right? And within it, there's all these little synaptic vesicles that carry acetylcholine. These will fuse with the cell membrane here, dump out the acetylcholine, which will diffuse on over the cleft, and bind to receptors here at the postsynaptic membrane. And from there, an action potential is reestablished and can stimulate the cell to contract. So we can see those steps here. Right? You can see the action potential, AP for action potential, traveling all the way down. Here, the action potential triggers these voltage-gated calcium channels to open up. This allows calcium ions in, and it's this specifically that triggers these synaptic vesicles to undergo exocytosis and dump or release the acetylcholine out of the cell into the synaptic cleft. That acetylcholine, since it's dumped over here, it diffuses from where there's a lot of it to where there's less of it on the other side of the cleft. There it binds to, see right here, the gated sodium channels of the sarcolemma. So this is the key that unlocks this gate. When it unlocks, sodium is able to diffuse into the cell. And sodium rushing into the cell is what caused the depolarization of an action potential, right? So all those positive ions coming in make the cell more positive, and an action potential occurs all over the muscle cell. When the action potential ceases, so when our neurons are no longer stimulating the muscle cell, or consciously stopping whatever movement we were engaging in, that means no more acetylcholine, right? So those gated sodium channels close back up. Sodium can no longer pass in, so the cell goes back to rest. The cell stops dumping out acetylcholine, but there's still some that remains in the cleft, right? That could keep signaling the postsynaptic cell here. To ensure that one stimulus equals one contraction, and muscle cells don't keep being stimulated once we're done, enzyme acetylcholine esterase breaks down acetylcholine into its component parts, acetic acid and choline. That choline is mostly reabsorbed to make new acetylcholine. So remember, action potential traveling down, like driving to the ferry terminal. When you can't go any further, it reaches the river. We gotta get on the ferry. You can think of calcium ions as the conductor. It's what's telling the ferry to go. The ferry is acetylcholine that takes the signal over the cleft, where the electric action potential can be reestablished. We can get back in our car and drive away. This is another image from an earlier edition of the book that breaks down the process in a little bit different way, a little bit simpler, fewer steps. The number of steps isn't important as long as you understand what's occurring here. So now we know how the muscle cell is stimulated, what occurs at the neuromuscular junction. We can finally talk about how that muscle cell contracts. We can put it all together in what can be called the sliding filament mechanism, as it relies on these filaments or myofilaments sliding over each other. So first, we'll see an action potential traveling down the axon of the motor neuron all the way to the presynaptic terminal, like we just talked about. This causes those voltage-gated calcium channels to open up and calcium ions enter, like we just talked about, the conductor. This triggers those synaptic vesicles to release the neurotransmitter acetylcholine into the cleft, the ferry, which then unlocks those sodium-gated channels on the postsynaptic membrane, the sarcolemma, which allows sodium to rush in, making the cell more positive, causing the depolarization of the action potential. So these are all the steps we just talked about, all news. Now what happens though? What does that action potential actually do in the muscle cell? Well, first it travels all over the sarcolemma, that exterior cell membrane. But remember, we had to call it the sarcolemma. It had a special name because it had this special feature. The T-tubules were transverse tubules. These were folds or tubes that traveled to the interior, the core of the muscle cell. Also recall that the T-tubules were closely associated with the sarcoplasmic reticulum. Remember, this was called a triad with two parts of the sarcoplasmic reticulum sandwiching a T-tubule. This action potential travels from T-tubule to sarcoplasmic reticulum, making the sarcoplasmic reticulum more permeable or leakier, which then causes all the calcium ions stored within it to float on out into the cytoplasm or sarcoplasm of the muscle cell. Now the calcium is freely floating along. It's going to bind to the troponin of the thin filament, which then causes tropomyosin to move as it changes shape. Remember, these were our co-parents that were really overprotective of their daughter actin. Calcium, in this case, was the distraction, right? So calcium binds to troponin, changes its shape, causes tropomyosin to move off the binding site of actin. So now myosin is free to attach to actin at that binding site. Now a cross bridge is formed, which is just a bridge between the myosin head and the actin, a bridge between the thick and the thin filament. Recall that these filaments make up most of the muscle cell. They're just packed in there. So as the calcium's floating within the sarcoplasm, it's just going to hit all these troponin molecules, since there's so many in there. Now, finally, it's the fun part, the power stroke. The myosin heads break down or hydrolyze ATP chemical energy, use that energy that's becomes available as ATP is hydrolyzed to move. The moving is pretty subtle. is just bending that myosin head towards the M line, the center of the sarcomere. Because the myosin head of the thick filament is attached to the actin of the thin filament is pulled over the thick filament towards the M line. As the thin filaments on each end of the sarcomere are pulled towards the M line, the whole sarcomere shortens. This is called the cross-bridge cycle, because it will keep going as long as there is enough ATP to fuel it, and as long as there's calcium there to keep troponin and tropomyosin off the attachment site, so actin and myosin can stay connected. When the stimulation ceases, so when we cease to want to contract that muscle and the neuron is no longer stimulating the muscle cell, it'll stop releasing acetylcholine. That enzyme we saw, acetylcholine esterase, will break down the acetylcholine in the cleft, leading to the ceasing of that action potential on the muscle cell. Calcium ions will be re-sequestered in the sarcoplasmic reticulum as they diffuse away from troponin. And with no more calcium, troponin returns back to its original shape, allowing tropomyosin to cover up that attachment site on actin again. Actin and myosin can no longer stick together, no more cross-bridge, no more contraction. So, putting it together, here we are at the neuromuscular junction, our action potential traveling down. When it reaches the end, or the axon terminal, it's going to cause these voltage-gated calcium channels to open up. Calcium ions coming in tell the synaptic vesicles to dump their acetylcholine out into the synaptic cleft. Acetylcholine, ACH, drifts over, diffuses over the cleft to bind to the gated sodium channels on the muscle cell. The gates open up. Sodium can rush into the muscle cell, makes the cell rapidly more positive, and an action potential develops here on the sarcolemma. It then travels all over the sarcolemma of the whole muscle cell, including those T-tubules. You can see here why those T-tubules are important, because they will carry that action potential down into the muscle cell. With no T-tubule, it would just remain on the outside. But it can go down the T-tubule where it will signal the sarcoplasmic reticulum to become leakier or more permeable. As it becomes leakier, all of the calcium ions stored within it are going to drift out into the sarcoplasm or cytoplasm of the muscle cell. And in the sarcoplasm, we have tons of myofibrils made up of myofilaments, thin and thick. So it's going to bump up against all the troponin on the thin filament. Binding to troponin causes it to change shape. That change in shape causes tropomyosin to move off of the attachment or binding site. and myosin can attach to actin. A cross bridge forms between the thick and thin filament. Myosin can then hydrolyze or break down our chemical energy of ATP, accessing that energy and using it for mechanical energy or movement. It's just a small movement, just a shift in orientation, bringing it closer to the M line. M line would be over here on the left side. Middle of the sarcomere. Because myosin is attached to actin, actin of the thin filament also moves towards the M line, pulling Z disc closer to Z disc. Right, so this is our M line in the middle, Z disc, zigzaggy, so it's easy to tell apart. So myosin is going to keep on attaching to actin and pulling it little by little, micron by micron, closer to the M line from the left and the right. Right, pulling, myosin pulls actin this way and this way. This causes the sarcomere to shorten. When all the sarcomeres are short, the whole myofibril shortens, the whole muscle cell shortens, the muscle organ shortens, and generates that contraction that pulls on the bones and moves the body. This is pretty wild. All of the movements we engage in throughout the day rely on this, these microscopic proteins breaking down ATP energy to move everything that we need to. One note that trips people up: calcium. Calcium is so important to this process because it's used twice, right? Here it's used within the muscle cell to bind to troponin and move tropomyosin out of the way so myosin and actin can stick together. It was our distraction, right? That distracted our co-parents, troponin and tropomyosin, as it's released from the sarcoplasmic reticulum. But calcium has another role to play here at the neuromuscular junction. It's calcium rushing into the axon terminal that allows acetylcholine to be released. So two important areas it's used in. And this is just a step-by-step explanation of what happens during contraction. Again, the number of steps is not important. You just need to understand the process.